We're going to start with Matt Wright's going to give a brief introduction to VCI, and then I'm going to do a live demo of actually going through a variant and how the interface works. So I think, Matt, if you want to go ahead, I think you could start. Looks like there's still some unassigned.
Yeah, sorry, I was having. Sorry about that, Stephen. Thanks for the introduction. I was having a problem unmuting myself, but I'm fine now. I'm past that first technical hurdle. So should we give 1 more minute for folks to transfer in? All right, how many have we got on here?
Quite a few. And Matt, make sure to sort of introduce yourself and the team since you guys didn't present earlier in the meeting.
Yeah.
And I can go back to the other room and just see how many people are still being manually assigned by Danielle.
It looks like there's still 28 that are
can you see it?
Yeah.
How do you see that, Stephen?
That's what I was wondering.
Oh, so there's like a little number next to breakout rooms that tells me how many people are unassigned. I guess, I don't know if it's because I'm one of the...
You're seeing something we don't see.
The magical button.
Now it's down to 25.
So should we get started, Stephen, because we've got limited time today. For the hour, we've got to go like a fair pace. So should I introduce myself? So you see the 5 people up on the screen here. So it's myself, Matt Wright, and Christine Preston. We're the product owners of the Variant Curation interface, the VCI. We're based at Stanford in the Stanford development team. We develop the resources. When you write to the help desk, it's Christine and I that are at the other end. We'll respond to any queries that you have if you need any help, like registering, logging in, or you have any technical difficulties. And also, if you write to that help desk, actually, Stephen Harris and Jenny Goldstein and Xi Liu are the stakeholder proxies for the VCI. If you've got any ideas or suggestions for future improvements, etc., or want to know anything about the variant curation process, then we'll forward those queries on to them as well. And they're on this call too. Like Stephen's doing the presentation, will be a live demo in a moment. Jenny and Xi and Christine are available via the chat at any time. So as I say, I'll provide information to get you started in the special test versions of the VCI that we've set up for you to use today, before Stephen runs through the variant curation example. We encourage you to follow along. We sent out instructions for signing in to you all via email, and these instructions are also displayed on the ClinGen website for this session. And actually, Christine, if you could post those into the Slack channel as well, not Slack channel, the chat for this session as well, that would be helpful so people can see that link. For those,
yeah, I'll do that.
Yeah, thank you. For those of you who've already followed these instructions and the slides and signed up, and you don't need to do anything right now, you can go straight to your designated test site and just sign in and wait for Stephen to present in a couple of minutes from now. Please don't register your email a second time. For anyone who hasn't signed up yet, these following slides are for you, and I'll run through the process quickly on how to sign up. Okay. So we set up a number of test sites for use just for this session today. Please feel free to experiment with curation in these sites however you wish, because all the data will be erased from them probably next month. So it doesn't matter what you put in there, you can have a play. You can actually also, if you want to, it's always perfectly fine to visit these sites in the next couple of weeks if you want to come back and retry any curation, etc. We've assigned you to a specific test site based on the first letter of your last name. For instance, my last name begins with W, and so I would use a T to Z site, which is the Curation Transfer 11 link. However, we've actually set it up so that you can access any of these sites. So if you have any problems accessing one of them, then please do to try and log into one of the others. It's also perfectly fine if you choose not to log into the test site right now and to instead watch Dr. Harris's presentation and then come back in and do it yourself later. It's entirely up to you how you want to use today. When you go to any of these test sites for the first time, you'll first need to create an account. In the top right hand corner, as you can see here, indicated with a big orange arrow, you'll find a login link. you'll need to click that link to proceed. If it's your first time signing up, then please click the Create Account link. You'll then be instructed to fill in your details, such as your email address. Please use the email address that you sent to us when you registered for the workshop, because we've already pre-registered those into the system for you. So it'll be quicker when you sign in. When you enter it, please use only lowercase letters. When you enter your desired password, there are a few rules shown here. It must have at least 8 characters, at least 1 uppercase letter, at least 1 lowercase letter, and at least 1 special character, and at least 1 number. So bear those in mind when you put your password in. Also, fill in your first and last name as you'd like them to be displayed within the VCI. Once you've done all that, you can just click Create Account. And then if you go to the bottom of that modal, if you click the Resend Code button, and within a minute, you should receive an email that contains your unique verification code. If you don't see the email, then please check your spam or trash folder to see if it's been misdirected there. You can also resend the code again, and if you click that button again, and it will also resend the code to you again within a minute. Once you have the code, then please return to the VCI, and this time confirm your sign up by entering the code you received into the confirmation code box and click the Confirm button. After following these steps, you'll now be signed up to use that test site. You would now be able to log in and begin using the site. Your username when signing in is your email address. We've placed the link to these slides into the chat box of this session if you need further help logging in. During the session today, if you require any technical help at all with using the VCI, then please use the chat function in the Zoom session to reach out to Dr. Christine Preston or myself. If you have any questions about variant curation, please use that same chat function to contact Dr. Jenny Goldstein and Dr. Ji Lu, who are also on hand today to answer your questions. So if you have any further questions following the session today, then please also reach out to us via our VCR help desk, and we'll respond to those as soon as we can. Now over to Stephen Harrison, who would lead you through an example in the VCI. Thanks, Stephen.
Was muted. Okay, hopefully you can hear me and see my screen. Oh, it's like you're muted, Matt. Are you talking?
Yeah. Oh, I was just saying, just introducing you, but it's okay. You can continue on.
Okay. Perfect. All right.
I'm muted again.
No problem. All right, so I've logged into the interface here. So I'm in another production, or sorry, in a demo instance of the interface today. And so I'm going to go through an example variant through the curation process. But there's a few things I wanted to point out kind of first on this home screen, just to familiarize yourself with what the interface looks like. So whenever you have an account within the interface, there's this option to have different affiliations. And what the affiliations mean is it really is about who else can edit a variant interpretation that you started. So every person who uses the interface gets an affiliation that is just their own. So this one is just mine, Stephen Harrison. I'm the only one that can edit or change anything on any interpretation I start under this account. So all of our different expert panels have their own affiliations. So, you know, a hearing loss one, lysosomal storage. So all these are different affiliations that I'm a part of. So they all will be listed here whenever I log in. So if you are interested in creating an affiliation for your lab or a group of people, you can reach out to the VCI group here, and we can create an affiliation for your group. And so whenever you start interpretation, you want to make sure you're under your correct affiliation, again, because that controls who else can edit it and see it. And so I want to, in this case, I'm going to start under just my own account here and not under any of the other expert groups or affiliations. Additionally, because this is a demo instance, it doesn't have all the production data, but typically I'd be able to then see a history here of all of the different variants that I've curated, and I'd be able to do searches. So if I wanted to go back in and look at a specific interpretation I've done, and I knew the C dot or P dot, I could search for it here and go directly to it. All right, so now I'm going to start with actually starting a variant curation. So to do so, I'm just going to go up here and click on New Variant Curation. And once you click on this, it gives you options of how to identify the variant that you want to go through a curation for. You could either provide a ClinVar variation ID or a ClinGen allele registry ID. And so this is a way of controlling to make sure that nomenclature and the variant expression is correct. So I'll show you an example of how to do both. So let's say that this is the variant that I want to interpret today. This is a missense variant in MYH7, this C.2167C to T. If this is the variant I want to now interpret within the VCI, what I need to provide is the variation ID number. So it is the number that's up here in this top blue part, variation ID, this 14095. Also, if you're in ClinVar, it's the number that is here at the end of the URL. So I'm going to copy this ID, go back here, type it in. I accidentally took the front part there, and click retrieve. And what this will do is it'll first run a check to make sure that that you got the ID you were looking for. So it's now telling me here's the name that it has in ClinVar, and then some of the other HGDX expressions. So I'm like, okay, yep, this is the variant I want. I could now continue with my curation. Now let's say you wanted to use the allele registry ID instead. And so how you could use that, and so thankfully the allele registry option is there because let's say you want to interpret a variant that isn't yet in ClinVar. So the interface is not limited to things that are only in ClinVar. You could also go register your variant in the allele registry to get an idea and be able to assess it here in the interface. So Kevin did one of the workshops this morning about using the allele registry, but briefly what I'll show is what you need is just an HGBS expression to put into the allele registry to get an ID. So I'm here on the allele registry homepage. I'm going to select that I'm going to start with an HGBS expression. I'm going to paste in this NM and C dot, and then click search. So this variant already exists in the allele registry. Otherwise, it would come up here and say, do you want to register it? And I could just click yes. But this is that variant I want to look for. It's that C.2167C to T. So the ID that I would need from the allele registry will start with a CA. So it's up here. It's this canonical allele identifier. So I'm going to take this CA ID, copy it. Oh, and then the top part of the Zoom is in the way for me to get back to the interface. Okay. I'm gonna plus in, click retrieve, and you'll see I'm essentially going to get the exact same information as if I had started with the ClinVar ID. So while this is loading, I'll point out that you don't need to worry about which ID you're starting with. Once you actually click save and view evidence, everything after that point is independent of which ID you started with. So if for any one given variant, if someone started with the ClinVar ID or someone started with the allele registry ID, it doesn't create two different interpretations because it knows this is all about the same variant. So you only have to provide one or the other. You don't have to worry about getting both or having it have any sort of downstream impacts. I just wanted to point out how to gather both in case you have a preference for using one of these sources versus the other. So now that I have this variant I want to interpret, I'm going to click on this Save and View Evidence button. Can't tell if it did it here. Maybe click it again. Okay, sorry. So now it's going to take me to actually go start the interpretation for this variant. So now that I've started it, I can see the name of the variant up here. There's some links to different resources up here at the top. And then if I scroll down here, there are different tabs for different types of evidence. But what you can see right now is I'm still in this evidence view. That means I can't actually start an interpretation. I'm only looking at evidence that's been provided. So if I go to this population tab, I can look at information from Nomad, but I can't start an assessment because I'm under this evidence view only. So to actually start my interpretation, I want to click this interpretation button. And the interface is going to look almost the same, but it's now going to add in the ability to add ACMG criteria codes. So we're still going to be all these same different tabs to look at the evidence, but now that ability to actually interpret them. So I'm going to click on interpretation, and it gives you then this disclosure about not entering any kind of protected health information or any identifiable information about patients. I'm going to click agree. All right, so you can see now essentially the screen is looking the same. We have all these different tabs of the different evidence types. We have this top part with different link outs, but now there's this middle section, this variant interpretation record. So we can see all the different ACMG criteria codes here, as well as this ongoing calculator that as I go through, it'll be calculating what my pathogenicity is based off the criteria I've selected. All the criteria codes here start with a white background because I haven't applied any of them yet. As I apply them, they'll change colors. These criteria are divvied out into these different tabs, which we'll go through. But also, if you ever want to go to just one specific criteria code, if you click on it, it would take you directly to the section where you would interpret that code. But instead, I'm going to just kind of start by going tab by tab and looking at the evidence that way, as opposed to clicking into specific criteria that'll take us there. All right, so we have these different tabs where all of our evidence is gathered and where we can go about this actual assessment process. So the basic information tab and this GeneCentric tab are more background information. There's not any criteria codes to assess on these. It's just for overall information about the variant. So starting with basic information, first it would list if anyone else in the variant curation interface has looked at this variant. Again, because I'm in a demo instance, it's not showing anything here, whereas I know for this variant, actually, an expert panel has looked at it within the interface. So typically I'd be able to see that interpretation here. Next, we go down and we have information from this variant in ClinVar. So it's showing overall what the aggregate review status is in ClinVar, the overall clinical significance, how many submissions, and when it was last evaluated. Then down below, we can see each individual interpretation that has been submitted to ClinVar. So this is similar to if you're in ClinVar and go to that bottom section where you can see that table of each submission. That's where this is gathered from. So it's showing each submitter's interpretation of this variant, as well as each one's submission ID. If you prefer to look at this variant in ClinVar instead, you could click out and go directly to ClinVar to look. One side note here is right now, we only are pulling in the germline interpretations of this variant into the VCI. So if you are looking at a variant that may have some somatic interpretations, those would not be seen here at this point. But if you click see data in ClinVar, you would go to ClinVar and you could see those interpretations there. So if we keep scrolling down, there's now information about the transcript. So which, in this case, there's only one NM, so there's not as many to show here, but it's showing what the primary transcript is in ClinVar, what exon I'm in, out of how many exons, and then also showing the RefSeq transcripts and ensemble transcripts. We've also added in this annotation that calls out if it is the main select transcript or not, and if it's the canonical transcript. Again, in this case, there's only one, so it's a little bit more obvious here, but if there were lots of transcripts, all of these would be listed, and still this annotation of which is the canonical and what your expression is on each one of these different transcripts, and the molecular consequence on each transcript. So this is helpful in case that the impact may be more severe on a different transcript. You'd be able to catch that at this point. And then down below, the expressions on different genomic coordinates for 37 and 38. So the only other tab I'm going to go through real quick that does not have criteria, which is just background information, is this GeneCentric tab. And this is where you can get information about the different constraint scores from EXAC, PLI for if it's loss of function intolerant or not, missense constraint scores, and then also link outs again to other resources, Uniprot, and then ClinVar or any other kind of link outs you might want to see just for other information about this gene. All right, so now we can actually start going through these different tabs that actually have evidence on it and going through how we would add different ACMG criteria. So I'm going to work from left to right here, so I'll start on the population tab. So whenever you're in one of these tabs that has criteria, you'll see what the ACMG criteria code is and then what its definition is, followed by selections of being able to apply this criteria or not. So I'll show how this works once we kind of decide for this allele frequency data, which criteria we want to select. So I'm going to scroll down here. So we bring in data from Nomad, and we have which version we're using right here. So for example, there is a version 3 of Nomad, which is only genomes that is not yet in the VCI. So this is calling out exactly which version we're using. There's also data from Exact here. And then also if it was in the page study, 1,000 genomes or ESP, we would see those different sources here as well. So when it comes time to actually assess this variant, I'm going to have to decide now, based on the allele frequency I'm seeing here, which of the criteria codes I think is most applicable. So in this case, this seems like a pretty rare, borderline, almost absent variant from within Nomad. It was found in 1 South Asian chromosome and 2 European chromosomes. So in this case, I think it's rare enough. It's 3 out of 251,000 alleles that I'm going to say I think absence is applicable here. And so what I'm going to do is now go back up to where these criteria are and select which criteria I think is applicable. So I'm going to take this PM2 absence. So if I click on this pull down here, I can see I have multiple options. I could say this criteria is met, so that I think the evidence is sufficient here. I could say it is not met because maybe I looked at the evidence and I want to say that the I don't think it's sufficient to say that I can apply this criteria, or I can modify the strength. So for every asymmetry criteria in the interface right now, it gives you the ability to modify the strength to all the different strength options in this case. So for this one, I may change the PM2 weight now to supporting instead of using it at the MET level, which would be a moderate strength level. So I'm going to click PM2 supporting. So I'm going to apply this code, but at a supporting strength level. And then it gives you a free text field for information. And I'm going to put something like, found in, alleles in Nomad. Or you could give, if you want to use one specific population, you could call out which population you're using. I'm going to put what the denominator was, just so that it's more clear which one I was using. And then I'm going to click save. So what will happen now is you can see this PM2 changed colors because I've now added this criteria. And then you can see in this calculator bar, it's now showing I still have no benign criteria selected, but I have one pathogenic supporting piece of evidence. And that's because I did apply PM2, but I moved it down to a supporting strength level. One other thing to call out here is you can see for this section, there's an OR between these 3 different criteria types. There's a few places you'll see this type of logic in the VCI. This is because for instances where the criteria are all about the same type of information, it is limited to say that not more than one of these can be met, because you shouldn't say that a variant is above 5% allele frequency, but also absent. So this limits just to prevent sort of human error of accidentally applying more than one of these. So if I had tried to say BA1 or BS1 was also met in addition to PM2, it would have then given me an error that only one of these could be met at this time. All right, so now I'm going to move to the next tab, which is variant type. And under variant type, there's now different sub tabs, again, based on my variant type. So the missense tab is the one we'll look at for this variant because I'm looking at let's look up at the top again, a missense variant. But I'll show briefly what's on the other tabs. So if we were looking at a loss of function variant, it would then give us the ability to look at the criteria code PVS1. And there are some link outs here to look at guidance from the ClinGen SVI group about when to apply PVS1 or not. If I was looking at a silent or intronic variant, it would give me this criteria code about silent variants and not impacting splicing. Or if I was looking at an in-frame indel, this is where I would look at the PM2 or PB3 criteria about in-frame indels and whether or not they're in a repetitive region. So again, because we're on a missense variant, I'll use the missense sub-tab here under variant type. So there's this first section up here about whether or not missense is a common cause of disease or whether missense is not the primary disease type and it's primarily truncating variants. So this PP2 or BP1. And then there's also criteria down here about the in silico tools. And sorry, and one thing I should have noted, I didn't call this out earlier, is there is no requirement in the VCI to have to change every criteria code to a met or not met. It's kind of up to how your workflow works as far as how you want to select the criteria, but you don't have to, for every single thing, mark it as met or not met. And so that's why on the previous tab, I didn't go change any other criteria to not met, because in this case, I'm just going to focus on some of the evidence that I'm applying. So in this case, if I'm unsure if either one of these really are applicable for this gene, I'm just going to leave them right now as not evaluated, because I don't have enough evidence to really make a good estimate if either one of these two criteria I think are applicable or not. So I'm going to scroll down here to look at the in silico tools. So we have the two criteria codes here about computational tools, either supporting a deleterious effect or no impact. If I scroll down further, you can now look at the different in silico predictions that are brought into the interface. So we have the Ravel predictor, which is a meta predictor, as well as others like SIFT, Polyphen, mutation assessor, and so on. For each, it is showing what the overall score range is for this tool as far as what the output could be, and then numerically what this variant was scored as using this in silico tool, and then what the suggested impact threshold is. And this is from each tool's definition. This isn't ClinGen deciding what the impact threshold is for any of these tools, but about what threshold for showing an impact or not. Then if you look over here to this actual sort of prediction column, you can see there's a one-letter code for many of these tools. To figure out what these one-letter codes mean, if you press this little I button next to prediction, it then gives you the this key here. So for SIFT, there was a T there, so it is tolerated. For Polyphen, if it was a D, it's probably damaging, and so on from here. And so you can be able to look at these different single letter codes here and try to figure out what the impact could be. And then there's also conservation scores down here, and then link outs to different splicing prediction tools that you could be using as well. And so for these, you have to click out to go use the tool. We aren't able to bring in the scores to the interface for splicing predictors at this point. So based on this, overall, it looks like the predictions are pretty mixed. So Ravel, it's at a 0.68, and the impact threshold is 0.75 for saying it's damaging. And looking at the tools, so this one is saying tolerated, damaging. It looks like it's a bit mixed. Some of the tools are showing an impact, some are not. So for these, I'm going to say that this is not met, that I don't think there is enough evidence to say it's a deleterious impact because the tools are somewhat conflicting. So I'm going to put results tools and then click save. So what happens now, as you can see if I scroll up again here, this PP3 now changed to this gray color because I looked at this code and I decided it was not applicable. And so I applied or I said PP3 was not met. Whereas if I applied it, it would change to more of a green background here. So another interesting, helpful part here on the missense tab is down here at the bottom, the other variants in the same codon. So these are the PM5 and PS1 criteria about whether or not there's another pathogenic change in this codon that either is from either the same amino acid change or maybe a different amino acid change. So to help determine if this criteria is applicable or not, there is a helpful indicator here that tells you how many other variants are found in ClinVar in this codon. This isn't saying that they are necessarily pathogenic or not, it's just a heads up about if there's anything else in this codon in ClinVar. So I'm going to click here to now go look at those. And I can see, so the interface had said that there were 2 other variants in this codon, and you can see my search result is giving me 3, and that's because it is also including the variant that I'm already assessing, which is the bottom variant here. This is the one that I'm already in the process of interpreting, so that's why we're seeing 3 results here. But you can see there is this other variant in the codon, this now arginine to glycine change that is pathogenic, I can see an expert panel has called it pathogenic, as well as many clinical labs have said it's pathogenic. So based on this, I think that now the PM5 rule could be applied based on the fact that there is this other pathogenic change in the codon. So what I'm going to do, I'm just going to copy the full name of the variant here, just so I know what this other variant is that I'm saying is pathogenic. I'm going to go back here to the interface, and for PM5, I'm going to say it is met, and it is because variant such and such I think because expert panel has called this other variant pathogenic, that I can think this is applicable. So I'm going to click save. And now you can see this PM5 changed colors because I've now added this criteria as well. All right. So I'm going to move on to our next tab, which is experimental. And this is where you would answer questions about if it's in a hotspot or if there are functional studies showing an impact or no impact for this variant. So for all of these, there's this ability to add in literature evidence that would support your assessment of if these criteria are applicable or not. So for this variant, I don't have any information to add to these, but this concept of adding the PubMed ID, I'll be able to show on the next tab when I have my case and segregation evidence. It's the same type of process of being able to add in a PubMed ID and then determining if that criteria is applicable or not. So again, in this case, if I don't have any evidence about experimental studies or if my variant's in a functional domain, I'm going to move on to my next tab, which is this case segregation. All right. So this is where actually the most amount of criteria are found is on this tab. Observed in healthy adults, case control studies, segregation data, de novo occurrences, cis-trans data, and on and on. So what I'm going to do here is show how to add a PubMed ID that may have evidence that supports any of these interpretations. So let's say I have evidence of segregation from a publication, and I want to now add in this so that I can assess if segregation data is available or not. So to add in my segregation data, I'm going to click my source. I have, in this case, I'm going to add it from my publication data. So I have a PubMed ID. And you can see there are other options here, and I'll show how to add for clinical lab data as well, and some of these other options later. But I'll start here first with PubMed ID, and then I'm going to click Add Evidence. And so I've already looked up this variant to find this PubMed ID. Entered it in, and it's going to return the title just to make sure that this is the paper you really meant. And this is, yes, looking at MYH7 gene hypertrophic cardiomyopathy. Yep, this is the paper that I wanted here. So I'm going to click Next. All right, so this is now, we see this screen where there's lots of different options of adding in different types of evidence. So it can be a little overwhelming when you first open it, but essentially what it's allowing is for this single PubMed ID, there could be different types of evidence in this paper. It may not just be segregation. Maybe I also want to call out case control data or de novo occurrences or anything else. So it gives you that ability for every different code to add in any counts. But because I started this under the segregation section, it's highlighting that in green just to kind of call out where I think I have evidence. But it does, again, give me the option to add in additional counts to any of these other fields. So let's say in this paper, there were 4 segregations where the variant had segregated with disease. So I'm going to put in, so I put in my 4, and then I'm going to add in a comment here that segregated with disease, HCM, and 4 members. And if the family had a, code in the paper or something like that, I could also call out which family it was. So this is all just a free text field here. So I'm going to click Submit. And now 2 different things have happened here to this page. First, under the segregation data section, I can see this paper that I added along with my notes about how many total counts for segregations and my overall comment about the segregation data from this paper. Additionally, if I scroll to the very top of this screen, I can now see this overall summary section that for each data source that you're going to be entering, you can look at the counts for the different evidence types. And then will be a sum across different sources. So in this case, I've only added the one paper. So this is all that's here right now. And I only added the segregation data. So that's all that would be appearing. But let's say in this paper, there also was a de novo occurrence. So if I click back here to edit it, so it's already showing I added in my segregation data. But I also want to say that there was a single de novo occurrence. And it was a case where they did not confirm the parental. So I'm going to add in a 1. And then I'm going to, oh, it's already here from when I entered it before, a de novo occurrence in a proband with HCM. So this is still for that same paper that had the segregation data, but also in a separate family, there was a de novo occurrence. So if I click submit, you can see, so I still have those counts for 4 segregations, but now I also have my 1 count for a de novo occurrence here. So I'm going to go back to the segregation data. So let's say in addition to this publication that had segregation evidence, that I also have now clinical lab information that I want to be able to enter, some unpublished information that I got from a lab. So I'm going to add clinical lab data, like add evidence. So it gives you the ability to add in the lab's name, as well as any kind of IDs or information if this interpretation is in ClinVar. So let's say, I'm just going to sort of pretend here that let's say it was GeneDx who had provided me with this information about additional segregation data. So I'm going to now say the lab name was GeneDx. It asks if I want to provide, oh, I don't know what I clicked there, sorry, it went away. GeneDx. It's going to ask for the lab ID or an SCV number. These are both optional, but in this case, this will help kind of downstream to be able to have some kind of tie to where this evidence had come from. So I'm going to provide just what the SCV number, and this is GeneDx's accession number for their interpretation of this variant. I'm going to add this in right here. If I wanted to add the lab's ID number from ClinVar or the GTR, what I can do is if I click on GeneDx or whoever the person is I want to submit or whose data I want to use, their ID is this number here under organization ID on their submitter page. So their organization ID is this number that starts here with a 2. Put this in. And again, these are optional. So if the lab is not submitted to ClinVar, you can still enter that they provided you with information. This just, again, helps more for tracking. Click next. And it gives me all the same fields I would as if I was gathering data from a publication. So I can add criteria into any of the different evidence types here. But again, it's going to highlight in green the code that I started with, which was segregation data. So let's say this clinical lab gave me a family that had 3 segregations, and I was able to confirm this is a different family than was in the paper. Google Chrome here is remembering things I have previously typed. Add my 3 here and click submit. So now I can see here under the segregation data, I have 2 different evidence sources. I have the data from GeneDx as well as this publication, 4 counts from one, 3 from the other. And additionally, if I scroll back up here to the very top, I can see I have my PubMed ID and a clinical lab data. And it is now summing across all my segregations. So I have 7 segregations total. So what I'm going to do now is, sorry, a lot of scrolling here. Go back to the segregation data. So since I have 7 segregations total, let's say in this case, I think I want to apply my segregation evidence. But in this case, I'm going to move it up to a strong strength level. And in my estimation, I'm going to do 7 segregations total. And I could even, if I want to call out further, 3 from GeneDx, or from publication or some other information that just kind of will help if I ever want to come back to this variant and remember where my counts had come from, but these will also already be here. So applying this one, perfect. And then also because I had added my single de novo occurrence down here, and this was all the de novo occurrences I had, in this case, I also now want to apply my de novo count here. I'm going to say met, and I'm going to put just one occurrence. All right, so you can see now PP1 has been added as well as my PM5. So, and for this variant, there actually is more publications, but I'm not going to go through all those right now, just so I can show some other parts of this. So you can see overall now, I still have not applied any benign criteria, but I have applied 1 pathogenic strong, 2 pieces of moderate pathogenic evidence, and 1 supporting. So with this calculation, it comes out as likely pathogenic at this point. At any point when you're doing your curation, you also have this ability to add in the disease for your assertion and inheritance. So let's say at this point, let's just pretend this is all the evidence I have, so I'm all done now, and I want to kind of finalize this interpretation. So to add in my disease, if I click this Add Disease button, it gives me the ability to now use a structured ontology from the Monarch initiative to be able to say which disease this variant is with regards to. So in this case, all my programs have had HCM. I know this gene causes HCM, so I want to actually add HCM, hypertrophic cardiomyopathy, as my disease here. So in order to get this ID, I'm going to go to the ontology lookup service, this link here, and what I need is this Mondo identifier. So it'll look like Mondo and then a number. So I'm going to click out here. This is the Mondo disease ontology. I'm going to add in hypertrophic cardiomyopathy and click search. I can see I'm getting multiple hits back here. This looks like the one I would maybe want. So I'm going to click on hypertrophic cardiomyopathy. And once you get into any record here, you can look at sort of the ontology about where it falls. In this case, I want to use kind of this higher level hypertrophic cardiomyopathy as the disease, as opposed to getting into the specific, hypertrophic cardiomyopathy type 3 or type 4. And I just want to say hypertrophic cardiomyopathy. So now again, what I need to be able to put into the VCI is the Mondo ID. And I can gather that by, it's down here where it says ID, Mondo, such and such. I'm going to take this. I also could gather it, I think it's up here near the top too, this in this part. I could copy it out of here if I needed to as well. Go back to the interface, add in Mondo, click retrieve. And yep, that is the ID for hypertrophic cardiomyopathy. I'm going to save. Okay, so now I can see up here I have added hypertrophic cardiomyopathy, and my pathogenicity is likely pathogenic. And I can also add in a mode of inheritance for my insertion. And so I'm going to say that this is autosomal dominant. You can see there's multiple options here, as well as additional descriptors if needed that you may want to, that you could add in. But in this case, I just think autosomal dominant is sufficient here. I'm going to click save. All right. So also at any point in this process, as you're going through adding your criteria, you could go to this overall view summary option. So if I go here, I'm going to kind of skip this top part really quick and scroll down. And what it's doing here is it's separating out all the different criteria I've looked at into the criteria that I've said is applicable, those that I have marked as not met, and then those that I have not done any assessment on. So it's kind of into these 3 different buckets. So again, there's 4 criteria I have applied. It is telling me if the definition of each one, if I've modified the strength of it, and if I did modify the strength, what is its new status? So PP1, I did apply, but I modified it to strong, whereas PM5, I used as is, which is at the moderate strength level. And so I can see kind of this overall status at this point, whereas PP3, I said was not met because of the conflicting results. So based on this information, I can now look at my evaluation summary. I can see my calculated pathogenicity at this point is likely pathogenic. There is an option here to be able to modify. And so what this could be used for is if there's times where, with some additional clinical judgment or insight, you want to change what the calculated pathogenicity is. This also will often come up for variants that have conflicting information, where you've applied evidence on the pathogenic side, but also the benign side. And so you want to be able to change the pathogenicity. So it doesn't give you that option, but it does require you to write an explanation. And then there's this evidence summary box. And this is where they often will put in that overall evidence summary about why this variant was interpreted the way it was. So if I go back, I'm just going to kind of cheat here and go back to the variant in ClinVar and just take one of these evidence summaries. This is actually a very long one for the variant, just to kind of show. So again, this is where you would put sort of that overall summary about, where your different pieces of evidence were coming from. And I'm going to click save. So at this point now I've moved, oh, sorry, it is, I have to agree that yes, now I'm going to move this to a provisional level and submit it as a provisional interpretation. So the different statuses in the interface is it starts as in progress until you decide you want to move it to now a provisional interpretation where it saves what you have at that point. After that, you could then move it on to being approved. So a lot of our expert panels will kind of start with an assessment, get all the way through, and then mark it as provisional until the expert panel weighs in on if they agree with the interpretation or not. And if you ever want to reassess the variant, all the previous interpretations would be saved here. So I know we are running out of time, so I won't go into this part too much, except say that, yes, you can log back in and change interpretations, and it overall will keep this record of all the previous interpretations you've had for this variant and whatnot. All right, so I think that is everything overall I wanted to cover. I can see that there have been some questions that have come into the chat, which I've not looked at. So I don't know if Matt or Christine or Jenny or G, if there's any questions that have come up that you think we should answer live, or if there's anyone on who wants to ask a question live. Oh, I can't hear anything. I don't know if maybe people are not.
It looks like we have one question about the difference between the demo and the production sites.
Yes, that's a great question. So the demo ones are for practice, as in nothing that you do in the demo is going to be saved long term. And so correct me if I'm wrong here, Christine, but I think the demo ones often will get wiped about every month. So we often will use the demo instances for training purposes, whereas the production interface is things you'd want to keep, and other people could, look at what your interpretations for that variant have been. So it's more for real interpretations as opposed to just practice or training.
Yeah, and I would just add to that, we are now posting on the top of the demo site when our next wipe will be. So you can see it's going to be in July. And we encourage people to have registration in both. There's a number of people that we've encountered who only want the demo site, but we certainly encourage anybody to register for both.
One thing I realized I should have noted, once you're able to move a variant to being approved, there's been this option to be able to take the data from the VCI and put it into a ClinVar submission form and submit it to ClinVar. So I know there was a question yesterday about if anything from the VCI automatically goes to ClinVar, and it does not. It is completely up to you as a user of a curator within the VCI about when you want to share any of your interpretations externally. So this is true of both the demo and production interface, that nothing will automatically be shared with ClinVar or externally. It's completely up to you when you want to push anything out.
So we do actually have a couple more questions. I was going to say, I can't be sure, but I think that this will, because we're the last session, I think we can go over by a few minutes and it won't cut out.
Yes.
So I think we do have time to answer these questions. But if we don't, people, then please reach out to us at the help desk if it cuts out. So the first one is, what is the difference between the pathogenicity calculator in the VCI and the pathogenicity calculator in the ClinGen allele registry?
Yes. So these are two different tools. The the VCI, the variant curation interface, is what our expert panels are using that to kind of go through this process. So there is also this other tool, this pathogenicity calculator, which is slightly different. It's another option to be able to assess the variance. But yes, we were focusing more on the variant curation interface today, as that's kind of more of our prime interface.
Yeah, and I think a lot of us, I don't know if there's anyone from the Baylor team on, I think it's been helpful for some groups that want to do more batch rule application, like transferring data from one system to another. It doesn't have all of the sort of ability to document in detail a lot of stuff like the VCI has, but I think some groups have done some more batch processing with data. I don't know, Matt, if you have a sense of what groups have been doing with that platform.
I don't know. We'd have to get someone from Baylor to comment on that, who they've interacted with on that. I don't know the answer to that. But I would say that the pathogenicity calculator within the VCI literally just tracks as you go along. Each time you make any evaluation, it tracks it calculates what the pathogenicity is. So it is just a tool to track your progress in terms of pathogenicity.
Yeah, I think you're right. Like if a group had already curated and marked all the codes in structured form, you could load it up to that site and it would just automatically calculate the pathogenicity. In fact, I think the CNV group is doing it just as a way to make sure that the codes and the points they're applying, a slightly different version of it for CNVs, just to add them up and make sure there's not like an error. We've found in the past that groups that are trying to manually track, I have a PM2, I have a PP1, and a PM6, so that equals X. They sometimes just make an error in aggregating, so that function of calculating it for you happens automatically in the VCI, and that is what sort of that smaller application does in the allele registry, is just calculate it, but it's not allowing you to bring in all the data, see it, document notes and rationale, change things, all of those additional features that VCI has.
This is Kevin from Baylor. Yeah, I agree with that assessment in terms of the.
Oh, thanks, Kevin.
Thanks.
No, that was good. That was very good. I was going to jump in if I heard anything incorrect. No, thank you.
Save me from myself.
Yeah.
So the next question, I can answer this one, Stephen. So the next question is, can you request for an affiliation for your group in the demo interface? So yes, we allow any group that wants to curate together as a group. Within the interfaces, you can set up an affiliation. Any group can do that. You just have to write to our help desk. When you set up an affiliation, it applies to both the production and demo, but if you're already using the demo, that's fine. It will just be set up in there too. So if you're interested in creating as a group, your institution can set up or lab can set up an affiliation, or if it could be a group of researchers across multiple sites that are all working on the same like disease, for instance, domain. We can set it up for whoever wants to set up an affiliation and just write into our help desk. And the next question is, how many prediction tools have to agree to decide on the variant? And does the number what matters, or the nature of the program?
Yeah, I think it does depend more on the nature. There's not necessarily a right or wrong answer here. So a lot of groups have moved to using more of a meta predictor because it is easier to just sort of look at one source as opposed to trying to look across multiple, because the more you look at, the more likely it is that they're not going to all agree if you keep adding in more and more. So yes, a lot of groups have been using more Ravel, using a meta predictor, or deciding for their gene, that the score from maybe SIFT and polyfen together is the best estimate of a damaging impact for their gene versus others. And so then they maybe will limit it to just looking at those two. So we do display all of these in here, but you could decide for your variant, to base it off just a single predictor, if that's what you think is more appropriate for the gene that you're looking at.
I didn't see any other questions. I don't know if anyone's got any questions, write them quickly into the chat now so that we can, because it hasn't cut out, so we've got a few minutes.
And one last thing I forgot to call out was if you go up here to the top, there is this help section that does have a link to the SOP for the VCI, as well as this help page for VCI, which is more about some of the technical help parts of the VCI, about like, how do you start the variant? You know, how do you click this, make this appear? And so this is a really good resource that if you have questions that you can go through about registering, you know, what's an affiliation, how to begin, all of that kind of information.
And I've got to jump off to another meeting, but I did want to repost MA's post around the evaluation survey for the conference. It will be sent out by email, but I'm just reposting a link to the evaluation survey. It's not anonymous because we do want to be able to follow up on useful suggestions from people that send in feedback, but we do appreciate all of that feedback.
All righty, well, thank you, everybody. Yeah, if you have any
there was
questions.
there was one other question in there. I've just put it. Can you generate a PDF variant report using the VCI?
Yes, so Matt, Christina, correct me if I'm wrong here, but I believe once you're on this summary page, there is the ability to get the PDF, and now I'm trying to remember how to do that because I have not often done that before. But there is a way, right, to export this as a PDF?
Yeah, so if you click on the provisional, if you scroll down, sorry, slightly. So if you go where it says provisional summary there next to the button, you can, so at all times we have a snapshot, like a summary of both all provisional and approved, like classifications. And so you can view that anytime. And if you go to the bottom there, you got print PDF there,
Yeah, perfect.
There, and you can choose,
Yeah, so in this case, I hadn't actually approved this one, but if there was also an approved one, I'd have the option to do it for the, approved. So yeah, you can click on this summary for any of these, and that's how you could get the PDF. Perfect.
So we will follow up by sending an email out to all the attendees to invite people to register for the production interface if they're interested in doing variant curation. And we'll provide you with further details as well for affiliations and everything else at the same time. But thank you very much for attending today.
Yes,
thank you, Stephen.
Yeah, no, thank you. If you have any questions, feel free to reach out.
Yeah.
All right. Thanks, everyone.